And next, we want to cover appropriate characterization methods. But before we do, I'd like to push out our next poll question, and I'll review those responses in just a minute here. But what I want to ask our panelists, how do purification methods, let's talk about that. How do they affect the effectiveness? Or why is characterization critical to process development? And Dr. Murray, can you kick us off?
Yes, for sure. I think that is, again, it just links back into making sure that your final product is as pure and reproducible as we were mentioning before. Making sure that, so in Organicell, what we have done is not only that we are measuring the size of those exosomes or those extracellular vesicles, let's say, and that our final product is an enriched product that has the smaller extracellular vesicles, quote unquote, exosomes, right? But also that the characterization of the surface markers are on point, which one more time corresponds with making sure that those are exosomes. And most importantly, what we are working in is making sure that the microRNA component and the proteomic component are the ones that are being analyzed every time. So we know what is our drug, what does our drug contain, and from that point, therefore, what are the ramifications or which pathways are we looking exactly into and the mechanism of action of our drug, and why is it that we're studying these diseases that we're studying. So I think characterization is one of the most important things. I think the whole medical and scientific community agrees in these extracellular vesicle components that that is the key of your product. That is the key of the reasoning on how and why are you using specific exosomes rather than others.
Sure. Nicolas, can you give us your thoughts on this?
Yes, with pleasure. So maybe to answer the question of characterization, I think, well, characterization is critical as well on your process development, because you want to make sure once you are scaling up this process, so moving to 10 liter, 100 liter, you want to keep the same consistency of your product. And for that, you need to have well characterized your product from the beginning. If you are transferring to a CMO or another partner, you want to make sure that you do not have any deviation of your new product. So spend time on this characterization parts, and maybe regarding purity and purification, I think that, exosomes or extracellular vesicles are quite polydisperse, and it's still debating in the literature how large and small vesicle can really have a true impact on the mechanism of action at the end. So if you change your membrane cutoff, you filtrate your product, the efficacy of the product at the end can be impacted. So I would suggest from really the beginning of the development to start straight to scalable purification method, to have a plan for downstream development, and to keep it for first in vivo proof of concept, because you may lose some activity when you will go to preclinical, regulatory preclinical if you change the process, or when you go to the clinics. So it's critical to have it well in mind from really the beginning.
Good. Okay. Benny, I'd also like to give you the opportunity to discuss.
Yeah, no, thank you, Erin. I think that, I think key here is really, what's certainly been the recipe for success at Codiak has been, over years now developing a very detailed kind of analytical set of tools that enable us to, because we're engineering exosome therapeutics, we can really generate and limit that heterogeneity that here Nicolas is talking to. We do not see that. We can actually, with a very kind of narrow margin, highly reproducibly characterize and demonstrate that we have a high quality product with high purity, potency, and overall quality. And I think that analytical kind of characterization set here is really what enables scalable manufacturing, not just for preclinical studies, but for clinical trials. And at Codiak, we're kind of set up for scalable commercial manufacturing. And that investment has really been very clear. My CEO usually kind of tells the story that the first ever kind of scientific employee at Codiak was really a head of CMC and manufacturing. And I think that talks to understanding very early on that the production and the characterization of these exosome therapeutics will eventually define the success and the ability of continuing to forward it into the clinic and eventually into a hopefully a commercially kind of important medicines. So I think that it's important for all steps, and if we don't pay a lot of attention to the characterization, we'll end up kind of spoiling the opportunity, because if we get too kind of dispersed products, you don't really know whether the drugs did work or whether it's kind of like an accidental finding. So, high reproducibility and limiting the heterogeneity, I think, is extremely important.